Hi everyone, this is Lecture 8, Prokaryotic Transcription. The material here is covered in Chapter 5 in your textbook, pages 83 through 86. Last time we talked about it, sort of an overview of transcription and the enzyme RNA polymerase. This time, however, we'll be specifically looking at prokaryotic beginning and ending of transcription and the mechanism. So let's get started. First off, a few quiz questions. The first one is this: RNA polymerase would make what RNA sequence from the DNA sequence below? The top strand is the coding strand, the bottom strand is the template strand, and all of the DNA is from the middle of the transcribed region of the gene. This sequence here, beginning AAA, GCTT, is the DNA, and your 5 answers are below. Question 2. RNA polymerase binds to what DNA region? Question 3. What is the function of the sigma subunit of RNA polymerase holoenzyme? Finally, 4. How does the inverted repeat sequence found in an mRNA transcript function in termination? We're going to cover 3 major things today: the initiation of transcription, the elongation of transcription, and the termination of transcription. These are the 3 major steps that I want you to know. These are from figures in your textbook in Chapter 5, Figure 5.4. It begins by showing transcription initiation. Transcription initiation begins with sigma factor in bacteria. Sigma factor is shown here, looks kind of like a telephone receiver in the old style. It particularly binds to 2 regions of the DNA, and it's color-coded green here. This is the promoter. The sigma subunit binds to a region called the -10 region. Some of you may know this as the TATA sequence, T-A-T-A. Sigma subunit also contacts the DNA in the -35 region, which is shown here. Then the yellow enzyme is the RNA polymerase. Together, the RNA polymerase and the sigma subunit are called the holoenzyme. Notice that the RNA polymerase's mouth, the catalytic portion, touches to the junction between the green and the pink DNA. This is to serve as an illustration of the difference between the promoter, which is shown in green, and the gene, which is shown in pink. We call this junction the +1 site, and that is where transcription begins and where the gene begins. Once the holoenzyme is bound, it begins untwisting the DNA at the promoter. RNA polymerase has its own helicase activity. It unwinds the DNA, breaking the hydrogen bonds to result in the open promoter complex. At that point then, new nucleoside triphosphates are added, beginning at the +1 site. It's labeled here as the initiating nucleotide. It has 3 phosphates because it is a nucleoside triphosphate. Once the promoter is established, once the open promoter complex is established, then RNA polymerase begins the actual process of transcribing the gene. This is the step known as elongation, and RNA polymerase travels again in the direction of from left to right in this diagram, and it synthesizes the new RNA, as we learned last time, in the 5' to 3' direction. In this diagram, the RNA is shown as a green strand. A portion of this RNA is hydrogen bonded to the template DNA, as you can see here. However, as the RNA polymerase elongates down the gene, the RNA is removed from the DNA template and is sent out the back of the RNA polymerase. The RNA polymerase then winds back up the DNA so that it reforms a double helix behind it. Sigma subunit also falls off during this point. Remember, this is just an initiation factor. But RNA polymerase will travel down the RNA. It reads the template in the 3' to 5' direction, synthesizing the new RNA in the 5' to 3' direction. 2 strands that I want to just tell you about here. We have the so-called template strand, which is the portion the RNA polymerase is reading. Here it's on the bottom. This is the template strand. The coding strand is the part on the top. The coding strand has the same sequence of bases as does the RNA, except that we switch U's for T's. But the coding strand ends up being the same strand sequence as the RNA. Elongation continues, as shown down here at the bottom, and the RNA gets longer and longer. Finally, in prokaryotes, RNA polymerase will terminate at a specific sequence, and for different genes, it's different sequences. However, there is a common theme among many bacterial genes, which is called a stem loop, or otherwise known as a hairpin. The way a stem loop works, this is the RNA sequence at the very end of the gene. It forms a region of localized complementarity, usually it's CG rich, as shown here. And there is ability for this RNA to fold up on itself so that internally hydrogen bonds. Remember, RNA is single-stranded, but it can fold up on itself to form internal hydrogen bonds. This loop, shown here in green, is not hydrogen bonded to itself, so it does not H-bond. Following this stem loop is a region of U's primarily. The U's, remember, have only 2 hydrogen bonds per base, and as this RNA would be bound to the A's in the DNA, this region serves as a destabilization between the RNA and the DNA, so that it makes it easier to unwind. This whole structure here, this stem loop, when it is formed, it interferes with the elongation of RNA polymerase. RNA polymerase begins to slow down and eventually falls off of the DNA. And that is what causes the termination. If you look at the stem loop sequence, shown up here on the top, you'll notice that there is a region of 2-fold symmetry, which is shown in pink. For example, 5' to 3', it's read A, G, C, 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 G, C. If you read the same sequence in the reverse direction, on the bottom strand, A, G, C, 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 G, C, it's the same, so that when it's created as an RNA, those 2 regions will form 2-fold symmetry and be complementary to one another. So there you have transcription initiation, elongation, and termination in bacteria. One more point. In bacteria and eukaryotes in general, in transcription in general, there are multiple RNA polymerases that transcribe a given gene at a single time. This allows you to create many RNAs from one gene, and thus amplify the transcribed signal. All right, thank you. See you in class.